Hello everyone, my name is Binfeng Xia. On behalf of Sanofi U.S., I would like to give a presentation for the FDA MIDD training group. The topic of this presentation today is application of physiologically based pharmacokinetic models in Eliglustat clinical development. These slides give an overview of Gaucher Disease Type 1 or GD1. Gaucher Disease Type 1 is a multisystemic lysosomal storage disorder resulting from acid beta-glucosidase deficiency, which is a rare disease with fewer than 200,000 cases per year in the U.S. The therapeutic approach for GD1 including enzyme replacement therapy or ERT or substrate reduction therapy or SRT. While SRT and ERT work in different ways, the goal of each treatment is the same: to reduce the accumulation of excess glucosylceramide or GL1. SRT inhibits glucosylceramide synthase, thereby slowing down systemic-wide accumulation of GL1, allowing the cell's residual enzyme activity to break down the substrate. Cerdelga or Eliglustat is a glucosylceramide synthase inhibitor indicated for the long-term treatment of adult GD1 patients who are CYP26 extensive metabolizers or EMs, or intermediate metabolizers or IMs, or poor metabolizers or PMs. These slides show the key ADME properties of Eliglustat. Eliglustat is highly permeable and highly soluble with high first-pass metabolism, result in low bioavailability of less than 5%. Eliglustat shows nonlinear and time-dependent pharmacokinetic profile and is mainly eliminated via metabolism with less than 1% of dose excreted unchanged. The most important aspect of Eliglustat properties is its metabolism via CYP26, a polymorphic enzyme, which is the major elimination pathway. The polymorphism of CYP26 can significantly affect the pharmacokinetics of the drugs which are CYP26 substrate, such as Eliglustat. 4 CYP26 phenotypes are defined. They are ultra-rapid metabolizer, which is also called UM, and extensive metabolizers, EM, intermediate metabolizers, IM, and poor metabolizers, PM. And the parent drug level could vary significantly based on the metabolizer status, resulting in either adverse drug reactions or no drug response. In addition to the CYP26 metabolic pathway, Eliglustat is also metabolized via CYP3A at the secondary route, as well as via other CYP-mediated pathways, including CYP1A2 and CYP2C19. And Eliglustat could be exclusively metabolized via CYP3A in patients with CYP26 PM phenotype. These slides introduce the critical modeling and simulation challenges for the Eliglustat. The complexity of Eliglustat drug metabolism resulted in significant challenges during the clinical development and submission. It was hoped that the application of PBPK models to assess the wide range of drug-drug interaction concern for a new compound used for a rare disease. And hopefully, PBPK model can help to simplify clinical development plan and subsequent labeling. Typically, a very large number of potential drug-drug interaction conditions were of interest, such as various permutation of CYP26 and CYP3 inhibitors, as well as CYP26 phenotypes. And traditionally, this would have been evaluated using dedicated phase 1 clinical DDI studies. However, it would have been difficult to enroll adequate CYP26 IMs and EM subjects in the DDI studies, and also conducting a large number of clinical DDI trials would be very expensive. So the question is if modeling and simulation approach such as PBPK modeling can be used instead of conducting clinical DDI studies. During the clinical development of Eliglustat, the modeling simulation played an important role to reduce the number of clinical studies, and only a few key studies were conducted as listed on the slide. Here we have 3 examples, which is Eliglustat co-administered with paroxetine, ketoconazole, and rifampin. And these key DDI studies were also important as a key anchoring dataset for the PBPK model verification. This slide introduced general modeling workflow for PBPK. As you can see from this slide, there are 4 different steps. The first step is to develop base PBPK models. The base model development including entering information of drug-specific and PK-specific informations. And both of them are elements of PBPK models. The drug-specific information including molecular weight, pKa, and other physical-chemical properties. The PK-specific information refers to relevant PK parameters, for example, the drug clearance and the volume of distribution. After we develop the base PBPK model, this model needs to be verified with Eliglustat observed data in clinical studies, including different CYP2D6 phenotypes. For example, whether our model can be predicted the PK of Eliglustat over time in CYP2D6, PM, and EM genotypes. Then, the model will further verify to predict PK of Eliglustat under known DDI conditions. And this was also done with the key anchoring DDI clinical trials, as I mentioned before. If the model can predict the DDI effect under known DDI conditions, the verified PBPK model was used to predict the DDIs of interest, which are currently not tested in clinic. And we will step through each of these model building steps in a little more details over the next few slides. So this slide shows the base model verifications. And here we use a data set from a phase 1 clinical studies where Eliglustat was given at 100 milligram dose. And the dosing regimen is we give a single dose on day 1 of Eliglustat, followed by BID dose from day 2 to day 8. And we conducted this study in both CYP2D6 poor metabolizer and extensive metabolizers. And from this graph below, the open circle represents the observed data of Eliglustat systemic concentration. The line in black represents the mean simulated systemic concentration of Eliglustat by the PBPK model, which we use a software called synthase. And the line in gray represents the simulation with multiple virtual trials building synthase. So as you can see, the model captured the observed data in both CYP2D6 PM and EM populations very well. So in this case, the base model was verified. And next step, we verify a model under known DDI conditions. Here we apply 2 DDI scenarios for the verification. First, the co-administration of Eliglustat at 100 milligram BID dose with paroxetine, a strong CYP2D6 inhibitor at 30 milligram QD in CYP2D6 EM healthy subjects. And second, the co-administration of Eliglustat 100 milligram BID dose with ketoconazole, a strong CYP3A4 inhibitor at 400 milligram QD in CYP2D6 EM subjects. And the first 192 hours in the graph showing below, we simulated the Eliglustat PK after repeated administration of Eliglustat alone. And the simulation predicts the PK profile of Eliglustat very well. The red arrow stands for the time of starting dose of the inhibitors. And you can see the simulated PK profile after the starting dose of inhibitors are increased. They also capture the observed data at C12 level and also the full PK profile at the last dose. Therefore, our model can be verified with known DDI conditions. In this case, the DDI effects of strong 2D6 and strong 3A inhibitors was predicted successfully. After the verification of base PBPK models and verification of models under selected DDI conditions, the model was further used to simulate untested DDR scenarios. The example here is to show the simulated DDI effect when dual CYP2D6 and CYP3A4 inhibitors were co-administered with Eliglustat. So in this graph, the solid line represents the Eliglustat simulated PK at 100 mg BID dose alone, and the dotted line represents the PK profile after Eliglustat was co-administered with these dual inhibitors, proxylene at 30 mg QD and ketoconazole at 400 mg QD in CYP2D6 EM subjects respectively. As you can see, the simulated PK profile with dual inhibitors are much higher than the PK profile with Eliglustat 100 mg BID alone. So which suggests that the DDI effect under dual inhibitors are very strong. So because of the previous modeling verification steps, the outcome of complex DDI situation on Eliglustat PK with multiple perpetrator drugs can now be predicted with high confidence in any CYP2D6 phenotype populations. As initially stated, the main motivation of this modeling and simulation work was to optimize the data usage and minimize the burden of unneeded drug-drug interaction studies. This slide summarizes the prediction of untested and clinically observed drug-drug interactions. The PBPK modeling predictions for clinically untested DDI conditions are highlighted in blue in this table. This included predictions in CYP2D6 IM and PM subjects, and also the DDI with moderate inhibitors. In addition, the effect of complex multi-drug interaction, for example, the co-administration of dual inhibitors with Eliglustat, can be also studied, and the potential risk can be further understood by the simulation. The listed simulations are only the major modeling simulation results, and other simulations, such as drug-drug interaction with weak CYP2D6 or 3 inhibitors, were also conducted to support decision-making or otherwise can be inferred from results of clinical DDI studies. Also, please note that this table only summarized predicted fold change in drug exposure, but the drug labeling recommendation was actually based on the actual drug exposure after co-administrating with perpetrators, and this should be considered also together with the safety threshold. This slide shows the dosing guidance information from the U.S. package insert of Cerdelga, and this is indeed a complicated dosing guidance with various DDI-related scenarios. Eliglustat alone is prescribed as 100 mg dose in its tartrate salt form, which is equivalent to 84 mg free base form, and the BID dose was prescribed to EM and IM subjects of CYP2D6, while the QD dose was prescribed to the PM patient. With co-administration of inhibitors, the dose interval of Eliglustat will be either once daily, not recommended, or contraindicated. The dose guidance highlighted in green was supported by the clinical DDI trials or other clinical studies, whereas all other drug-drug interaction scenarios were supported by PBPK modeling. Therefore, modeling and simulation has provided significant support on the dosing guidance for Eliglustat and can be used as a classic case example for the application of modeling and simulation to support clinical development of new chemical entities with complicated ADME properties. To conclude my presentations, we already see the impact of modeling simulations on Eliglustat clinical development. The modeling simulation support had significant input into the Eliglustat package insert and also reduced the number of DDI studies conducted. In addition, the modeling simulation gave us insight into DDI potentials and the concomitant therapies that would never have been tested in classic DDI studies. And that's all for my presentations. Thanks all for your attention. Bye-bye.